Thanks. So in this view, there is clearly DNA's information. It started with RNA. Today, DNA is the information, the code, the library. RNA is an intermediate step. And proteins are the machinery of life that do everything in the cell. Is there any dent in that picture? RNA is an intermediate step.
Virus started in some time.
Is a virus live? By the way, this is one of those things that is complicated, but the traditional explanation of a virus is not because it is not self-reproducing. It needs some host. I'm sort of escaping to answer your point. Now back to the eukaryote. You don't see any dent in the picture. So let's look at the RNA as an intermediate step. Ribosomes. They contain RNA, and they have, in fact, enzymatic activity. The ribosomes has proteins and RNA, but RNA is there. It's not an intermediate step. It's part of a big construct, of a big machinery. You may have heard about sRNAs, RNAis, short RNA. What are they known for? You haven't heard of those? So.
They have regulatory functions.
Yes, so they interfere somehow with proteins. They have regulatory functions, and it is clear. But you're not here.
I wrote my name down on the.
Joanna. Hello, Anne is. So that's the red hair. Oh, I'm sorry. and this is your glasses, Andrea, right?
Here.
Oh, you, Andrea. Oh, yes, that's Maria. Oh, yeah, now I get it. So I need pictures of the two of you. Unfortunately, I don't have my phone on me. Could you bring me a stick? Or a photo on a stick next time? Thanks. So they are regulatory, but regulatory means that's a strict violation of an intermediate step, right? RNA is not mRNA that does a protein. This is active RNA. It sits somewhere and sort of blocks some reaction or activates some reaction. And there are, in fact, many of those known. Now, there are, in the ribosomes, there are RNAs that are more than 100 nucleotides long. Typically here, these sRNAs, RNAis, they are sort of 20. So they are much shorter. Some of them are 6 nucleotides. They are much shorter bits and pieces. Now, if I asked you, how many 100 and longer do we find in human? Not how many copies, but how many different non-coding RNAs. So some are in the ribosomes. Do we have 10? Do we have 100? Anybody has any idea? So I'm not talking about these short ones. Of these we have a lot. Now, but they are sometimes so short that to call it identity is complicated. So you have 10 mer, and then you have one nucleotide different. Is that the same or is it different? For the ones that are 100, you can clearly say, 99% sequence identity called the same. So the surprising reality, and that was one of those big papers now 11 years ago from the Phantom Consortium in Japan, in which in a single paper with 143 authors, they found for mouse 180,000 transcripts, about 16,000 protein coding. 5,000 of these were new. But the surprising reality is they found about 20,000 non-coding longer than 100 nucleotides. 20,000. So we have as many of these intermediate things that are not intermediate as we have proteins. Why do we need so many? And what do they do? 11 years later, we still don't know. When that paper was published, that was a complete surprise. Nobody had anticipated anything in that, anything near this order of magnitude. If they had published 1,000, people would not have believed it. But again, so for the time being, I'm going to follow this sort of central idea: DNA, RNA, and protein is the machinery of life. Protein function comes on different levels. You know that function has all kinds of aspects. Let's start somewhere simple. What is a protein? How do you define a protein? Ever heard that problem?
Sequence of amino acids in stable form.
Is sequence of an amino acid what?
In stable form.
So you take a sequence of an amino acid, such as this one, and you say that whatever stable form. What do you mean by stable form?
Basically, like it has like optimum energy state.
Oh, it folds.
It folds.
Oh, that's too far. That's too far. So part of the protein may be post-processed, may be cut. So maybe the protein that actually folds is minus the first 50 residues or something like that. We'd still call that the protein. That's a post-translationally modified site. We'll talk about that. So that will be cleaved off, cut off. That only will fold, but this is still part of that protein. But the question is, what is part of that protein? So typically the idea is you have a gene that is spliced together from different exons. Right? You combine exons, one gene, one protein. But that is not quite true. That's an oversimplification, and I will get back into that for the ATPase. But in principle, once we have protein, your idea is right. In principle, the thing that you throw into solvent that adopts a unique three-dimensional structure, that protein leads to the unique structure. The idea, the experiments come from infants and then show that a protein can adopt a unique three-dimensional structure on its own without needing any agent. There are different types of protein structures, and they aggregate to larger things. And these larger things, so I'm not sure, I believe this slide is wrong. These larger things, here's an example of the ATPase synthase that in fact converts electrochemical energy or potential into mechanical energy. So there's a gradient here of ions. It pumps natrium out, potassium in, 3 natrium out, 2 potassium in. That gradient leads to this turning around, this starch. The starch turns the whole mushroom up there around, and that creates that energy. The whole thing comes with this. The point now is you have different elements here, different parts. Typically we would refer to them as domains. You see them in different colors. For the ATPase, they come from 4 different chromosomes. That is one protein. This is not a protein complex. We perceive this as one protein. But this one protein actually is spliced together from very different regions in the genome. So this idea a gene is a protein is completely violated here. So what I'm trying to say here is even the idea of a protein is not that simple. Okay? One operational definition is what is coming from the same origin in the genome, introns spliced out, exons together, is a protein. Again, there are exceptions to that. Now, once we have a protein, a sequence such as this one here, we find out about function often by, in fact, aligning it against the database. In the database, we find a protein cytochrome P450. We say this, in fact, is also cytochrome P450. Now, how can you define protein function? You can define protein structure by saying it's a coordinate in space. How could you define protein function? And I believe in the exercises you already have come across some definitions, right? What definitions could you name?
A chemical reaction, it catalyzes.
So you can essentially classify all the proteins according to the reactions they catalyze.
Not necessarily, but it's a subgroup of proteins which catalyze
reactions.
reactions.
And then you essentially you give every reaction, every sort of chemical reaction gets a code, and that code is inherited by the protein. That would be one classification. And in fact, we come back to this. Let me just look at this example here. That's an alcohol dehydrogenase, ADH5 it happens to be, from human. And that is an enzyme, and that is in fact catalyzing exactly such a reaction. It breaks down alcohol. So that's exactly the answer. It's one of these answers. There's no formal answer here. But there's already something else that you see. For instance, here you see the structure. And looking at the structure, you may or may not see that what you see is actually a dimer. It's a homodimer. So you have two copies of the same thing. In this particular case, that's biological. What that means is that if I removed one by some artificial way, it could not function as an ADH. It could not catalyze that reaction. In order to catalyze that reaction, it has to be a dimer. Okay? Only as a dimer it functions. Now, the fact that it has to be a dimer, bless you, is not really its function. But it has to do with its function. Right? It's some aspects of its function is that it has to dimerize. Now, when you get down to a little bit more of a detail, there is an enzyme, there's a sort of a classification of enzymes. We will get back to that. That's the number here. Then it says that it is part of a metabolism of fructose. And it says many, many other things. Now, this first line here is back again to your reaction catalysis. And that is essentially the enzyme classification. In the enzyme classification scheme, you have a hierarchy. The first level is an oxyreductase, and the second level you get into more detail. I'll get back to that in a minute. And then ultimately we get to exactly the reaction you talked about. That's the enzyme that has that reaction. But there's something else that is important in what I put up before. It is part of the glycolysis pathway. In this pathway is that one here. This is one of, I don't know how many, but every blue point here is one enzyme. Every circle is another protein. The arrows are reactions. There are many, 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 many proteins in this glycolysis reaction pathway. And being part of this pathway, not only being the alcohol dehydrogenase, but the alcohol dehydrogenase in that pathway, is another important part of its role, its function, right? So we see that there are different levels of function. There are different aspects. One level, this chemical reaction, for that class of enzymes, I can formalize. The belonging to a pathway, I could also formalize, but maybe not all of them I can formalize. And in fact, if we looked at a more abstract level, where the chemical level is how an atom is bound, biochemical level is essentially a transferase, cellular level is a cell cycle, it's part of cell cycle, developmental is time, the time when it's regulated, physiological, for instance, is related to disease, genetic is dominant or recessive. What is your interest in biology? What is your focus? Determines on what you see as the protein function. They are all very different aspects on very different levels of fine-grainedness, so to speak. Mostly you talk to cell biologists or biochemists somewhere here. So they care less. Structural biologists would care more about that level. Biochemists would care more about this level. But geneticists would care about these levels. And none of them is more right than the other. Okay? So function in some sense is one way of saying that is everything that happens to or through a protein. Now this happens typically, I said that sequence determines structure, but sequence determines structure in a dynamic sense. We need to allow motion, so the scaffold is only part of allowing motion. Motion is relevant for function. And here is one example for the type of motion that happens upon a substrate binding here. And you see that the substrate, essentially, so the wobble that you see of the protein is essentially Brownian motion. The substrate is in this Brownian motion pulled typically by something such as a force field, and it samples different spaces. And what we see essentially is an overlay over a Brownian stochastic process of sampling. and an energy or a potential-driven process of, in fact, sampling more often in a certain part of space, and then, in fact, at some point locking into the final conformation. And now you see that motion of the protein is relevant because it allows essentially the substrate to be passed on, to move on. On a different example here where we have receptors, you see how the Brownian motion happens, how in fact holding the ions in the gate, blocking them in this particular case here, and you can imagine how this Brownian motion will ultimately lead to a situation where the ions are pushed. Right? And again, we have motion here. You see that these two helices, or 4 helices, are moving with respect to each other. So you have motion on different levels here. And these different levels are important for keeping this in the right ratio. So can we predict protein structure from sequence? Generally we cannot do it because it's complicated. But for very short proteins we somehow can do some time scale. And one of the most successful, or the one that goes for the highest time scale, is a machine called ANTON. This is a special purpose machine, a very big parallel machine standing in New York and built by David Shaw, or David Shaw Enterprise. I'm actually not sure.
Is it the guy from D. Shaw?
That is the guy from D. Shaw, yes. Yeah, that's the guy from D. Shaw. Mm-hmm. And so that simulates protein dynamics on a previously anticipated level. And I just want to show you a brief snippet of a movie that shows how you can sort of do drug design with ANTON. On the next one.
The water molecules, and in fact it's a very snug fit. This is when it finds this binding site, you can consider the water is fitting very neatly in there, which is what the drug really wants to do, among other things. So here's another one. This is also binding to SAR kinase. It's a drug called dasatinib, and it is an actual drug that's approved for leukemia. And it's known again where that binds, and this is another experiment he did where he just threw it into the box. It's harder than that, but put it in the box, and it's fairly quickly in this case found the rough right area. It's going to pause here, and it's actually kind of interesting because, well, maybe it already did it. A little groove opened up here, and it got distracted by it for a second, but then eventually it goes to exactly where it's supposed to. And then this is a different kinase. It's the epidermal growth factor receptor, which is it's actually a family of 4 different proteins that are implicated in several different indications. But in this case, for this drug, the drug is approved for the treatment of breast cancer. It's called lopatinib, and I guess it's hidden right now. This is the last one I'll show, but the drug's going to search around again, and it's going to find the right place. And again, there are a few false places that it is before it finds the right one. One very interesting thing about this is, this isn't like a new finding, but it's what's called induced fit. In the process of having the drug molecule fit into a protein target, it's not just that the drug itself moves around, it's also that the protein moves around, making space for it. And the induced fit part is when the drug actually gets there, you can actually change the shape of the protein locally. And many methods to computationally identify the drug binding site and look at how well it actually works as a drug assume a rigid protein. They might actually start with several different snapshots of the way the protein could be, and then what's called dock all of these little drug molecules into it, see where they could fit, not through the actual dynamics, but just kind of moving around like this in all possible rotations. But there's some kinds of drug binding which probably occur in areas that aren't even pockets. They're not, usually these kind of drugs bind to little depressions, and they fit very nicely, and they have certain types of chemical interactions. But in some cases, we don't think they're there. This has been postulated. We're seeing some examples that I haven't shown where here's create its own pocket where you couldn't agree to where it was going to bind and then bind in there. But this is all very, very preliminary, intriguing from the viewpoint of drug design, but early stage. So switching to computer science for a sec, ANTON was designed by a classic.
So again, they need ANTON to do this because ANTON has such a power that it allows you to give up on the idea of rigid body. So you do not have to assume that the protein is stable when the drug comes, but you can actually let the protein wobble around and be changed according to the drug coming in. Now this simply means that you have to sample a much larger set of conformations. So this simply means you need a much larger computer. And the only way they could do that is by essentially building a very big parallel machine that is optimized for this type of problem. And back again to your question, this is D. Shaw research, has nothing to do with a hedge fund.
I think it's like his,
other than
it's his science venture because he does it for fun, I heard.
So for a function you need motion, and sometimes, still motion we believe is part of ordinary ordered structure. So you still have to adopt a rigid or the same three-dimensional backbone skeleton. Upon this skeleton you can move this one a little bit around, and this one moves more than that one maybe, and all of that is part of the structure. Okay? That is all the idea of structure determines function. Comes disorder. In disorder you have this little red part here that binds to this blue part, and upon binding you, for instance, he talked about induced fit, David, upon binding you induce a secondary structure for the red one. But if you now, at this state, in the binding state, short lighted, if you could do that, you can't, but if you short lighted it, now the red would always be the same at different time points. If you short light at that point, 2 different time points, the red would look completely different. So here we have a thing that is, as they call it, disordered. And there's a functional reason for it being disordered, so that it can sample more conformations. And in fact, there's a lot of disorder, in particular in eukaryotes. And we believe that disorder has something to do with the complexity of an organism. Here are 2 examples of disorder binding. This one binds DNA, 2 different types of disorder. So a lot in eukaryotes. Now let's go back to metrics for protein function. Again, for sequence we have matrices such as E-value, pairwise sequence identity. For structure we have RMSD, so you simply look at the differences of 3D coordinates. For function, 1 is again the enzymatic classification. You have this hierarchy of enzymatic classification. The generalization of that is the GO, gene ontology, and that I had hoped you would say. You have all heard about GO, no? Yeah. So why did nobody say that? Because it was so obvious? Essentially, if you think about it, GO is an extension of the enzymatic classification. Enzyme is reaction-based for a particular type of reaction, and GO tries to do the same thing for all the proteins. Now, for all the proteins, they realized very early on we cannot sort of have one scheme. We have one scheme where we look at the process. So catalysis is the analogy in the ADH story. Molecular function, that now is the enzymatic activity, alkyl dehydrogenase, and the cellular component. Where is this function happening? Localization. So there are 4 different trees here. DNA metabolism as a biological process, then the molecular function of the DNA binding domain, and the cellular component of this protein being in the nucleus. Now, the first question is, structure is conserved in sequence. We know that already. Similar sequence essentially gives a similar structure. That's what we talked about last semester. So everything above this line here, so percentage sequence identity is here, number of residues aligned is here. Blue points have similar structure, red points have dissimilar structure. The curve, the black curve, somehow shows a line where mostly things above that black curve are blue. So mostly things above the black curve have similar structure. We can use that for homology modeling of structures. That is the best way we can predict structures. Let's talk about some asymmetry and exceptions in this. Similar sequence does not always have a similar function, and a similar function does not always have a similar sequence. Same sequence we have here, different tissue, different function. How can that happen?
Post-transcription modification.
That is one perfect example. So it could be that the protein, in fact, there's something that happens to it. You could put a sugar upon it, and somehow in the other environment that is, say, in a secreted environment, you have more sugar, and then suddenly something cannot happen. Binding, for instance, sugar is positively charged, binding DNA could be an immediate example where this would have an influence. Yes. What else could it be? In this particular case that I'm talking about here, it's not the case.
DNA methylation?
Yeah, that somehow, again, you would argue is a post-translation modification in some sense, right? But here, the issue is much simpler, or worse is not the right word. So we have essentially two different tissues have slightly different pH values. Slightly different pH values lead to the protein to have a different way of electromechanical potential, static potential. I'm sorry. And that, in fact, induces a different function. Okay? Different environment. It could also have been that it binds to something else, that something has to set it on. This is like post-translational modification, but it could be that in some environment it is bound to some protein, and in some other environment it's not. And depending on whether it's bound to a protein or not, it could do something else. And that again happens. Now, here's an example where we have two very different chymotrypsin left, subtillin on the right. We have two different structures that essentially do the same thing. You would typically talk about converge to doing the same function. The domain problem underneath that we will encounter during the course of this lecture again and again is that in the database of annotations Swissprot, so the database of all protein sequences is UniProt. And UniProt has about 80 million, 85 million at this point. Swissprot is a set of proteins for which we have every protein in here. There's some experimentalist who has done an experiment on that and has published something about what this protein does. And that is about 0.5 million. And how do these proteins get annotated? Essentially by a team of annotators. Right? How do you picture this? How many people annotate this? 1? 2?
Should be like media, right? Should be like a media professor sitting at the table. Like
no, the professors are too expensive. No, so we have at some point Swissprot has had about 100 annotators. We're not all full-time working, but it is a large annotation team that is essentially working full-time. And there is a variety of backgrounds of these. In fact, some of them are retired, and some of them are on the level of professor. Yes, but it's a mixture of people. Essentially, they read the literature. That's the way it happens, right? That's it. Now, Swissprot typically has an annotation of something that says, for instance, ATP sulfurylase here. And then there's an annotation for this one here. There's a synthase. The alignment is the red region. Now we get a region in this one for a protein that has no known function. What is its function? So the problem that we have here is we do not know whether this annotation really is for that region or local. We do not know whether the synthase annotation for which region that was. So in particular, we do not know whether this in fact implies that they have some similarity before we know a little bit more about the enzymes, and whether that may be reasonable or not. We don't know whether the annotation is that region here, or whether that is yet a different one that is not in any of these two, right? that's one of the big complications, because most annotations, in fact, are not on that level of detail. You already had an exposure in the exercises to moonlighting proteins, to proteins that have more than one function. In some cases it is a binding of a formation of a complex, a cofactor. There are all kinds of different ways in which this moonlighting can be induced. Any idea why it's called moonlighting?
It's moonlighting that you make someone believe something which is actually true.
Okay, ultimately we are through today, and that is good, because then we get to the next lecture. The next part here is about homology-based inference and what we can do. Now, let me skip this part here. The general challenge for homology-based inference has to do with how much of the evolutionary track record we can reproduce and what the underlying image is. So let's begin with this first question. How do you define speciation, if you haven't been in my last lecture? Anybody? Nope. So typically speciation means that you start with a common species. The common species somehow splits. The split is such that afterwards the two different species, they cannot have offsprings together. Okay? They can mate but not have offsprings. The way we could imagine this is we have some flies that like this type of food or environment or light, there's some that like this one, that is the way they mate. You know, these that love bananas go to the banana, disaster comes, the banana is washed to the other side, and we create on two sides of the river essentially two different communities that have two different preferences in terms of their food. Even if now the banana came back to the old island, they still would not cross-mate. Can that sort of thing happen? I mean, it's a sort of cartoon here. Is that reality? In fact, you can do that with flies in the experiment. You simply have two different feeding mechanisms, and after not that many generations, 8 or more, so this is just days essentially, or a few weeks, you get different mating preferences, and you can create not quite the speciation events, but you can create subspecies or groups that behave very differently. How that then translates to whether these in fact are same species that can interbreed, or they are different species or not, so the details are a little bit mixed. Getting back to the level of the protein, one issue here is when we talk about homology on the level of the protein, again here's sort of a functional classification, what we ideally like to say is the ADH1 in human, the alcohol dehydrogenase 1, the other one that I showed you was 5, and the one in plants that are somehow related, and there somehow is a common ancestor. And that's the idea behind the word homology. Homology originally goes back to the idea of homology, similarity in corresponding body parts of two organisms that come from the same ancestor. There's an evolutionary idea here. In this particular case, homology, again, down to the level of proteins, implies exactly that. There's a common ancestor, and they are all offsprings from that common ancestor, right? Now, homologs in principle have a common ancestor. The statement that there is a common ancestor could be like this, or it could be like this. Both of those are common ancestors. The tree in between is not defined, because in order to define the tree in between, we may need to have a history that we cannot access easily. Now, typically, we talk about ADH1 and ADH1 in human and in plant, in this particular case, as orthologs. Where ortholog essentially means, well, okay, we have, now when the two species split, they have essentially the same genome. Right? And each of those, in this particular case, would have an ADH1. Since they essentially have the same genome, they're essentially the same cell types, they essentially do the same thing. Okay? So when I see something like that, I can infer, if I knew the function for plant, I could infer that the human protein also has that function, simply because there's an evolutionary connection. Not because they are similar in sequence, but because that happened that way, all right? It didn't happen that way on the level of the protein. It happened on that way on the level of the organism. But the protein is in some sense an imprint. In fact, it does happen on the level of the protein because you have reshuffling of exons according to their functional relevance. So then the tree is no longer there, but the rationale is still there. Now, assume you had a duplication event at some point. So you have, rather than one copy of ADH1, you have two. Since the second copy is no longer needed, you're free to diverge in function. ADH1 is doing its function, and you need that. ADH2 can adopt a new function. Right? That's the idea behind it, parlock. And we're not always sure that really we can trace the common ancestor, and that is one of the problems in these words, homologs, ortholog, paralogs. Over the course of my lecture, I'm trying to, again, I present them here, so typically the way they are seen. The problem is that the tree is very uncertain, and it's getting more complicated on the level of protein because of a variety of issues. Problem number one, genes and proteins are not reproduced. Domains are decoupled. This is the issue that I talked about before. And then the third problem is moonlighting. So there are many issues why tracing the evolution for a protein is not quite an easy thing to do. What we can look at is identity and similarity, and that is what we're going to look at. So in looking at identity and similarity between proteins, what we want to do is we want to look similarity in sequence to infer function. How well does it work? So say I knew that this protein is a methyltransferase. And now I find a protein at 99% sequence identity. In fact, I don't know it's a methyltransferase. And in this particular case I do. But if I wouldn't know and I would just read off that name, I would be right. So if I stopped here and I said everything that I accept is above 66%, you would see that 2 and 3 of the findings would be right, where right is simply saying methyltransferase, right? That is the second part comes in a minute. So if I go further down, I have 3 and 8 right, and 4 and 4 coverage. What I mean is, of all the methyltransferases I could have found, so there were 1, 2, 3, 4. So my counting is off a little bit here. So this is 2 and 3. And this is 3 and 3. So this is wrong. Oh, God. These are the kinds of things. This is elementarily wrong, right? And you have no idea how many lectures I've given in which nobody noticed that. Okay. So the question now becomes about a choice. So one case, again, my numbers are wrong, just imagine they were right here. You have 66% accuracy at 50% coverage, and another one you have 38% accuracy at 100% coverage. And the question is, what is better?
It's like what we want to do with that.
It's very, very important what you want to do with that. Now, we want to infer that proteins are enzymes in this particular case. So homology-based inference. So you know what you want. So
it goes like into meta level, like what do you want to do with homology, for example?
good. So I can. Okay, let's begin with a drug. You want to find a potential drug target.
I will do some statistics, for example.
So no, but assume that what you wanted to do is what do you mean by statistics?
I mean, like, since I'm trying to figure out like drug target, there will like some cost per protein. I will see like optimize in a way that minimize.
Pandu fully accepted what you say. So there are a couple of things you can look at, and ultimately it's down to you can define it as in dollars spent on doing putting a product into the lab, for instance. It's a fine way of doing it. But there's something else. Say, for the sake of the argument, this would not be methyltransferase. this would be the protein binds DNA or not. Could you now tell me that one of them is better than the other? Or DNA, again, is not a good example, but the protein is also not a good example. The protein, so we're looking at a particular residue, and I'm asking, is that sequence variant have an effect? And I believe now I'm getting to an example that I can defend. So my prediction is effect, not. I have binary classification. Those are my numbers. Can you now tell me? Why?
Because the baseline is 50%.
Exactly. So this is totally nonsense, right? So if this is another thing that is very important for you to realize. Any question such as this, ask yourself what is random. What's the background? What's the expectation, right? And one expectation would be if it's a 50-50 odd and you get 38, well, it may sort of be intellectually challenging to find out how can you be so much worse than random. And again, so you're smiling. We all have done. Anybody who has been in this field long enough has come up with a method that was worse than random. I'm sure I have. And so this is sort of challenging an experience that when you see this, it doesn't really make you laugh. But typically that comes after many years of investment, right? And but anyway, so that is one way of putting it. This is the clear decision. This is worse, okay? We still don't know how good that is. We know 66 is better than 50, but this is clearly not acceptable, right? Now, in order to make decisions like that, we need to look at other aspects, at other methods, and random is one of them. Now, let's look at one particular challenge. We want to see how well homology-based inference of function does for the example of predicting subcellular localization. And that is work that goes back to something that Rajesh Nair did, who's now at the FDA in Washington. Looking at a eukaryotic cell, we see a lot of subcompartments. In the simplified version, we have nucleus, a cytoplasm inside of the cell, and an outside of a cell. Now, if I wanted to, in fact, use homology or sequence relations to infer subcellular localization on this level of 3 states, okay? I know that when two proteins are very, very, very similar, they will have the same subcellular localization. And when they diverge, I will lose the signal. I expect that this leads to a situation where the accuracy of my method, so inferring by homology, goes down, while the coverage goes up. Coverage essentially means, so I want to predict that a protein is nuclear. When I only look at the proteins that are 95% sequence identity, the likelihood that I find things that have an experimental annotation is lower. The further I go, the more I will find. That's simple, right? How the curves go up and down, we don't quite know. They will go somehow up, right? So how can I now find out what reality is? How do I design my experiment? What do I need? So I want to check how well does homology-based inference predict subcellular localization. What do I need? Step 1. I need a data set. What data set? Step 2, open window. Wake up. So I need a data set of proteins of known localization. Swissprot has these. You have to be careful in Swissprot because many of those say by similarity, by homology. So those you can, so nuclear by homology. Essentially, they assume that homology modeling or homology inference works, and they infer by homology. We cannot use that to test how well it works, right? So there's a record that says by experiment. That's what we need to know. We need to take. So now we're not going to take the entire set. What we need to take, since databases are biased, we're going to carve out an unbiased subset, sequence unique subset, and then we're going to blast the sequence unique subset against everything. So if we want to avoid bias, we could do sequence unique against sequence unique, but why can we not do that? well, we wouldn't have any finding, right? We want to see how well can I use homology-based inference, and here is no pair of where you have any homology, so bye-bye birdie, right? 0. So we need to accept some bias. So I take the sequence unique subset and align it against everything else. Last semester I talked about similarity in structure. This semester about similarity in localization here. And that's the result. So number of residues aligned, percentage sequence identity. Blue, every pair in blue has the same subset localization. Every pair in red has different subset localization. Now what I'd like to do is to simply go through the room, assume that this is your finding in your thesis, is a graph that you see, and you want to use that graph for your publication, and you're in the results section. In the results section you essentially describe what you see. And I want everybody to have one observation. Just start from that around.
The blue ones were the ones with
same subset localization.
Same subcellular localization.
Yeah, every pair, so every dot is a pair, and the blues have
I'd say the more residues aligned, the higher the percentage of pairwise sequence.
So the more residues aligned, so you mean that there's a higher density of points up here? Yeah. Okay. So essentially, he's looking at this cluster here. Mm-hmm. Yeah, great.
Okay. There seems to be. It looks like two trends are there actually. Like.
Give only one. So okay. Everybody wants to observe something.
That many times there is same subcellular localization when few residues are aligned, and there is a high sequence identity.
So there's a lot of blue here, is what you're saying.
No, but there is a trend like the more the higher number of aligned residues and means a lower pairwise sequence identity. Like there's.
So there are more points here.
Yeah.
So there are more points.
Especially around 100. There is like between 100 and 200.
So there's a high density down here.
No, 160. It starts there, and then down to 330.
So I'm getting a little. Don't observe too much. A simple observation, but you're saying that there's a higher density on this side than here? I'm losing your observation now.
No, there is like a trend when we go from 100 aligned residues towards 300.
Yes, then?
This area, the pairwise sequence identity falls from like.
So in this area, you have more.
It falls from 100 when.
Are you talking about the gray line?
No, about blue dots.
So you have more blue dots in that area, or what are you saying?
No, the
work on it. Think it through and work on it. Make a statement. Yeah?
Do you repeat what the red dots were exactly?
Yes, that is. So blue is they have the same subset localization, red is they do not.
Well, if you don't have the same subcellular localization, it is like what one would expect, then their sequence identity is quite low. And if they have a high sequence identity, then it would be more likely that they have the same subcellular localization.
That's what we expect. Is that what we see?
Mostly, I would say.
So you see, so you say, forget about this axis. I hear you say, and you say that the axis is not meaningful at all. All we need to look at is a slice like that, and somewhere in this slice like that, I have a lot of blue. is what you say.
Yeah, well, there are exceptions, but...
Yeah, but that's what you say? Fair enough. Yeah? Crane, you jump in anytime you're ready.
Yeah, okay, so it's a bit difficult to explain. It's like there seems to be for observations between 100 and 300 aligned residues. There seems to be sort of a correlation between higher pairwise sequence identity and lower number of residues aligned seem to be correlated or blue dots, blue dots.
So you say something about blue dots.
Yes.
What is it about blue dots?
More blue dots.
Above some line?
Blue dots form not a line, but they form a
cluster?
A trend.
So the blue. So are you saying that there's something special with the blue, something that is here in this interval that is not true in this interval?
Yes.
And what is that something?
In the interval on the right, there are more. There is more blue dot. The majority of blue dots with high pairwise sequence identity.
Yeah, that's unfortunately that observation is already given.
Below 300 it goes like this.
Oh, so you're talking about some density that you see here?
Yes.
Ah.
It's very difficult to explain it.
Yeah, fair enough. Fair enough. That's much easier for me. Because I can point. I did not allow you to do this. Yeah, that's an interesting point. So he says there seems to be a high, so there's a density, so before we heard there's a high density here of Stefan, and now Quirin says, okay, there's another high density here in blue. Okay.
Yes.
Next. Simon.
I think there is no real like structure. The blue dots are everywhere.
Blue dots are everywhere. And in fact, the story is slightly more complicated. As you may already guess, there are blue dots underneath. Okay? Next.
Yeah, I have like a question. The gray line, is it just like for reference? Or
you know, the gray line is just the gray line was such that it was a good separation for structure between the red and blue. For structure, it was a good separation.
Okay.
And I just show the same curve.
Okay. Yeah. So what I was going to say is that the red dots, like they show a more or less a clear trend, but there is no real separation. Like the red dots, they go up to 60% sequence identity, and then more or less independent of the number of residues aligned, they're still red. So there is still some.
But you say something about the curve or not? So the red somehow fit to the curve, or is that something you said?
Yeah, like it.
Okay, fair enough.
Yeah.
We do want to get around. Anna.
Honestly, the two things I could say were just that. I'll try another one. The number of residues aligned doesn't seem to have a huge impact on the. It's a prediction, is it? Or is it the?
No, that's an observation.
That's the observation.
That's the real data.
Well, for me it seems that the number of residues doesn't... There's no correlation between...
Well, Maria says that there's somehow the red somehow follows that shape. Are you trying to negate that with the entry of saying no correlation? What then is no correlation?
Actually, I wanted to say that the blue dots are kind of evenly distributed throughout.
Evenly distributed throughout?
Yeah.
Okay, fair enough.
But it's sort of,
let's just take it there. Andrea.
There are no data points below
Great point.
10.
Thank you.
10
Yes,
residues of lines, and there are no points below,
Yes.
I think 15 or
So
10.
there's something white here. It's a very, very important observation. That we tend, again, so one of the reasons why I do this here is because typically in doing this, you all become aware of what we actually look at. You're forced to become aware what blue and red really means. But one is that this is what you have to do with your own plots. And you have to sort of look at them in a different way. That there's a lot of white is an important observation. It may not have a meaning, but first you have to put it down and then see whether it has a meaning or not. Pandu.
First, I would like to ask one question. Are both plots coming from the same set of protein? Can't be the case, right?
This is a different data set.
All right. So, well, what I realized, like basically, I mean, compared to the right one, basically, there's like less, I would say, clear cut line between sequence similarity and subcellular.
So here the fit is better than there, is what you're saying.
That cut is better because, like, you see, like, on the upper part there is no red at all, and the red part, like, there are proteins with actually very high.
No red at all is not quite true, but yeah.
I'm looking at the right part, like, there are a lot of red dots, even though they have like very high sequence similarity. Like, for example, they say almost 90%, but then.
Yeah, that is what I don't understand. That nobody has made that statement yet. There is red up here. Yeah.
Because the gray line is not a fit for this.
No.
For the other one.
Yeah. And fit is yes. I believe yes is the right answer to your question. No, it has nothing to do with this. And that is essentially what we were trying to find out. Julian. Yeah.
There's a trend that the more residues you actually can align, or for you still have the quality of alignment you take as layer point, I think, the more likely it's they are in the same subcellular localization.
Yeah, so they're more blue when you go in that direction.
The red is more on the right side, yes. I think blue might be even.
So let's look at a couple of things here. So this one here somehow indicates that we still have too much noise or too much bias in here. We should not have clusters. This essentially indicates that there are a lot of proteins that are entirely identical, almost entirely identical, 95% or more, over the entire range of the protein. This should have been excluded. We don't know how to exclude that, but when we observe this in the results, maybe we say we don't know a mechanism by which, so just artificially excluding 95% is not really biologically motivated. 90% is not biologically motivated. Since I don't know a number, I keep them in and I say, okay, here's a flag, we make a mistake. I don't know how to clean it up. Then there is this issue. I would contend that somehow you said that we can completely ignore this one here. I would contend that this is not quite true. So that there is some sort of trend indicated by this curve. It may not be a fit, and it clearly is not a fit as good as that. But in some sense, the most important point, I believe, ultimately are these here. And now we get to the issue. If you see a point like that in your graph, 400 residues aligned, 95% sequence identity, red, different subcellular localization, what's the first thing you do? Check your code, check your annotation. Typically, this would be a mistake. Very, very often. It may even be a mistake in the database. So spotting a mistake in the database means you have to really get back to the original literature and look it up. Maybe there's something that went wrong. In this particular example, they are not. They are real data points. And we will get back to that. Yeah?
The localization isn't unique for a protein. Do you have multiple annotations for the same protein?
Yes, you can. And in fact, proteins are wanderers in that sense. The meaning that they are all expressed on the ribosomes, and then they wander to different places. And some proteins are more wanderers than others. But arguably, there is what you may call a native compartment in which they stay more often. And so let me just make an extreme point that will come out the end of, in 6 lectures from now, when I will show you that we can predict subcellular localization, and if subcellular localization would not have a dominant feature in sequence, we could never do that. We can actually do that at a relatively high level of accuracy. So that, I believe, is the proof that there is a dominant subcellular localization. There's no better proof that you can have. But some proteins are wondrous and are more wondrous than others. And that is still giving us a lot of headache, that we cannot quite distinguish those.
But I guess why we have so these high red side.
They are not, that is not the answer here.
Yeah.
So I'll show you the answer. What the answer is, let's see. It will take maybe not next week. Okay, now the other issue that we all came across had to do with something like density. So the question of, is density increasing here? Are there clusters? Corinne had a cluster here. Are there clusters in that direction? Are there clusters in that direction? We all saw these. We all saw density of blue points. But actually we don't quite see that, the density. In order to see the density, what do we have to do? How do we have to replot it? Essentially what we have to do, yeah?
Well, you would have to zoom into the dense parts.
That's one way of doing it. So this is true. You could zoom into that. But that would still not give you an overall count, right?
Three-dimensional.
Three-dimensional. That typically goes wrong, actually. Remarkably often it's difficult to see in 3-dimension. So you could overlay distributions. So you could look at a blue and red distribution as 2 curves that simply sum over this and sum, so you could put them on the y-axis and the x-axis. That's one way. But ultimately, what we want to do, remember our goal. Our goal is to establish that we can use homology-based inference to predict subcellular localization. Meaning that we are looking for the threshold at which we can actually do that. We want to find out at a given level of sequence similarity, how well can we do that? And there are different thresholds I'm going to show you. One threshold is simply using this curve and looking at distances from this curve. So moving this curve in that direction, so the distance is computed here on the x-axis, not the y-axis, sorry. This is plus and this is minus. So moving the curve below, the curve is at 0, minus, plus values. Another way of looking at it is simply percentage sequence identities. Essentially what you suggested, to completely get rid of this axis and simply count according to percentage sequence identity. And the other one is e-values. Everybody clear about e-values, what they are? Anybody has an issue? No. So Psi-BLAST evals in this particular case. Let's begin with percentage sequence identity. Percentage sequence identity, I show you 2 curves. So this is 100. That is accuracy, meaning of all the ones that I say at that threshold, say I put myself at 80%, and I say that everything more than 80% sequence identical is the same subcellular localization. If I did that, then, I don't know, whatever the number is here, 95% of those would be right. Of all the subcellular localizations I could have found, at this level, I get roughly 50% or something like that, right? So this is the coverage. Coverage goes up, accuracy goes down. Okay? Now I see the same thing for the log E value, minus log E value from Psi-BLAST, and I see the same for the distance from the HSSP curve that or HVAL. The curve, the gray curve that I just showed you. Okay? And for all of those, you see, you can clearly infer subcellular localization, so there's a fairly high level of accuracy for some threshold. Which one of those is the best? What do you have to do in order to answer that question? What you have to do is you have to find a comparable threshold in the coverage. Say, that's one way of doing it. You could do it the other way around. Let's just say, so this is the axis here for coverage, 0.5 is here. So I want 50% of all the proteins I could have found. I want to find them. I find them at this threshold of sequence identity, 40%. At that level, percentage sequence identity, the accuracy is about 76 or 77. Okay? To find 50%, using percentage sequence identity threshold, I have 75% accuracy. Now let's look at the 50% point here. The 50% point here is here. And you see, now we are getting to an accuracy level that is close to 90. If you redid that for a couple of other points just to make sure, you would see that the e-value is clearly better than percentage sequence identity, right? Now let's just complete the story here, 0.5, and you see that this is even better. So indeed, Maria, you were the one who sort of observed on the one before that essentially, the animation, but essentially there is, although this is not anywhere near a fit, but there is still some behavior here. And that is clearly visible in the fact that this, at any level of accuracy that you put yourself, or any level of coverage you put yourself, it is above the others. You could also compare it directly, but that is one way in which it is. There still is a region here where, in fact, you have not 100%, and that ultimately is because in different tissues you could have different functions. And so, in terms of subcellular localization, you could have different subcellular localization. Again, I get into more detail for why that is the case. And the next step then will be to redo the same thing here for not subcellular localization. But how well can we do homology-based inference for enzymatic activity? And that will be next Tuesday. Thank you for your attention.